Now taking up another very important gland associated with our digestive system, and that is liver. Now liver, as we know, is the largest gland of the body, but it should not be confused the largest organ. Largest organ is considered to be skin, and it is located on the right side of the abdomen just below the diaphragm, and the liver is having number of smaller structures called lobules, and these lobules are surrounded by a connective tissue sheath called Glisson's capsule. Now if we see the structure of the liver, this is, we can see how the liver appears, and it is just underneath the diaphragm. This is the diaphragm, and as we can see, the liver is having two lobes. Now this larger lobe here, this one is the right lobe, and this smaller one is the left lobe. And these two lobes, they are joined by a ligament, and that is named falciform ligament. Then here, this green structure that we see here, this is the gallbladder, in which the bile juice is temporarily stored. And as we can see, this is the bile duct, and this bile duct is joining with the pancreatic duct to form the hepatopancreatic duct. Now at this region where the hepatopancreatic duct is entering into the wall of the duodenum, there is a valve, and the purpose of the valve is that it is regulating how much bile and pancreatic juice can pass into the duodenum, and this valve is the sphincter of Oddi. So both bile juice and pancreatic juice will flow in a regulated manner because of this sphincter of Oddi. Then this right lobe is further divisible into three lobes, and those are named right lobe proper, caudate and quadrate. So this right lobe is divisible into three, and these are right lobe proper, caudate and quadrate. Now if we take a section through the liver, so we will find that there are number of these pentagonal or hexagonal or maybe heptagonal lobules are there, and they are just arranged like honeycomb, and each hepatic lobule is a complete structure in itself, and it is taken as a structural and functional unit of the liver. Now liver is one organ which is having a dual supply of blood. Liver receives two sources of blood, and that is one from hepatic artery and the other is from hepatic portal vein. And each of this hepatic artery and the portal vein, they are giving out branches which are supplying to each lobule as well. So the liver is having a dual supply, and so does each lobule also get a dual supply. Now in this case, as we can see, hepatic artery is bringing oxygenated blood. That means it will be having oxygen needed for the functioning of the liver cells, while hepatic portal vein is bringing deoxygenated blood. But this blood is rich in nutrients. Now, hepatic portal vein is a vein which begins from the various regions of the alimentary canal in which the end products of digestion have been absorbed. So, this blood, although it is deoxygenated, but it is rich in nutrients. Now, both these hepatic artery and hepatic portal vein, their branches can be seen at the vertex of each of this hepatic lobule, and they are then leading into the interior of this lobule where they are forming sinusoids. So, that means both of these are opening into, like capillaries, there are special structures called sinusoids. Now, sinusoids are very similar to capillaries, but the only difference is that their endothelium, that is the single layer of simple squamous epithelium that the capillaries have, that endothelium is incomplete. So, as a result, sinusoids are more permeable than capillaries, and these sinusoids are then ultimately opening into a vein that is called central vein. So, this is how the entire circulation can be seen within the hepatic lobule. Now, if we take one region from this hepatic lobule and magnify it, so what we will see here. Now, in this case, we can see this is the branch of hepatic artery. And here this is the branch of hepatic portal vein. And both of these, they are opening into the sinusoid, which is then ultimately draining the blood into this central vein. Now, in this hepatic lobule, two important cells are there. One is these rectangular cells that we can see, these are the hepatocytes. And these hepatocytes have number of functions, the secretion of the bile, synthesis of urea, all those functions are carried out by these liver cells or the hepatocytes. And there are cells which are lining this sinusoids, which cannot be very clearly seen here, but we can mention them, and those are the Kupffer cells. Kupffer cells. These cells are on the lining of sinusoids. And these Kupffer cells, these are phagocytic cells. That means if certain microorganisms have been able to enter into the bloodstream, that means they were present in the food, and from the intestine they have entered into the bloodstream, but once they reach into the liver, because the blood which is directing away from, which is moving away from the intestine, that will be first carried to the liver. And in the liver, all those pathogens will be phagocytosed and the blood would be free now to circulate in the rest of the body. Now, coming on to the functions of the liver. So, the functions here, there are a number of functions, and these are just a few which have been listed out here. It secretes bile juice, then the red blood cells, which we know they are without nucleus and they have a short lifespan of just 100 to 120 days, and those are then ultimately destroyed in the liver. Then another function here is that they detoxify the blood by removing certain waste or poisonous substances. Then they store iron and vitamins like A, D, E, K and B12. The other functions, they make the plasma protein, various proteins which are present in our blood plasma like albumins, fibrinogen, and these all proteins they will be synthesizing from the amino acids Then they store glucose as glycogen, and this process is called, when glycogen is synthesized, it is called glycogenesis. Glycogenesis. And the reverse, whenever the blood requires or the body requires more glucose, that glycogen will be broken down to glucose, and that process is called glycogenolysis. Then of course it produces urea. So whatever excess amino acids are there, those will be broken down to urea, which would be a waste which is ultimately eliminated by the kidneys. Then it synthesizes and breaks down the cholesterol. Now after going through the functions of liver, let us see the constituents of bile juice. So here we have the constituents where, of course, just like any other secretion, there is water. Then there is sodium bicarbonate, giving it the alkaline nature. There are bile salts, and these are two of them, that is sodium taurocholate and sodium glycocholate. Now this is the most important aspect of bile juice, because these bile salts are the ones responsible for the emulsification of fats, and without emulsification the fats cannot be digested. Then bile pigments, that is bilirubin and biliverdin, and these are the products of breakdown of the RBCs, and in the RBCs it is the hemoglobin which breaks down to form a waste, and that is the bilirubin and biliverdin, in which bilirubin is in more quantity, and because of this the bile juice has a golden yellow color. Then there is some cholesterol and few phospholipids as well in the bile juice. Now the pH of this bile juice when it comes out of the liver, that is the hepatic bile, it is 8.6. But when it is stored in the gallbladder, that pH is reduced to 7.4, because when it is stored in the gallbladder, it is concentrated as well as it is acidified. There is addition of some hydrogen ions in it, so the pH decreases, although it still remains alkaline. And from the gallbladder there is a cystic duct, and this cystic duct will carry this bile or transport this bile into the bile duct. Now we can utilize this information and solve a question. So here we have, which of the following guards the opening of hepatopancreatic duct into the duodenum? Now this we had seen in the anatomy, that at the hepatopancreatic duct there is a sphincter which is named sphincter of Oddi. So this one is our correct answer. While with the digestive system, we have also studied ileocecal valve, that is on the junction of ileum and cecum, pyloric valve or the pyloric sphincter, that is from the pyloric stomach to the duodenum. Semilunar valve of course is part of the heart, which we will be studying later. Now after this we can carry on with a small discussion on the digestion process. After completing with the anatomy of the digestive system, now we move on to the physiology of digestion. And if we talk about digestion, it is both mechanical and chemical process. So mechanical is just where when we chew the food, we break it down into smaller fragments, but when the bonds are broken, then that becomes a chemical process, and that is an enzymatic process. So the digestion starts in our mouth itself, and mouth is specialized for ingestion and for beginning the digestive process. Ingestion involves mastication, that is chewing of the food by the teeth, and that is part of the mechanical process. And then this chewed food is swallowed, and that is called deglutition. Now deglutition is a reflex which is controlled by medulla oblongata. So when we are chewing the food, the tongue is continuously rolling it backward, and once it comes in contact with the soft palate, there are nerve impulses going to the medulla, causing the contraction of the muscles in the buccal cavity, so that our larynx moves up, the soft palate closes the internal nostril, and the only opening left out will be the esophageal opening, that is the gullet. and during this time when the larynx moves up, the epiglottis is flung in such a way that it covers the opening of the windpipe, and this process takes less than 2 seconds, but it is an important part because it is preventing the food from going into the nasal cavity or into the windpipe. Now, in the mouth, there is another thing that is happening is there is salivary amylase is acting on the starch, and the starch that we have taken out of that, nearly 30% of it is hydrolyzed by the action of salivary amylase. And salivary amylase is activated by chloride ions. It works at a pH of 6.8, that is the effective pH for it or the optimum pH for it, and starch is broken down to disaccharides, that is maltose and isomaltose, and a oligosaccharide, that is limit dextrins. So this process is taking place in the buccal cavity. Now this amylase, however, is not present in all mammals. It is absent in case of cattle, buffaloes, and also in case of carnivores. Now the food which is mixed with the saliva, that is called bolus, and this bolus is conducted into the pharynx and then into the esophagus, where by peristaltic motion it ultimately reaches into the stomach. Now next part of the digestion will take place in the stomach. So here food remains in the stomach for 4 to 5 hours. So stomach is temporarily storing that food as well, and this food mixes with the gastric juice to form the chyme. Now in the stomach there are protein digesting enzymes, pepsinogen and renin, and these are activated by HCl. So HCl is converting pepsinogen to pepsin and prorenin to renin. Renin is highly specific on which type of protein it acts, and that is milk protein casein. So the digestion of the milk protein begins because of the action of renin, then it will be further hydrolyzed by pepsin. And pepsin converts proteins into smaller peptides which are termed proteoses and peptones. Now we know that proteins are made up of polypeptides, and these polypeptides are long chains having number of amino acids. So when these long chains are broken up into smaller chains, those are proteoses and peptones. But certainly the protein digestion is partial. It has not been completed here. It would be completed in the small intestine. Now from the stomach, this acidic chyme with partially digested proteins will be passing on to the next part of the alimentary canal, that is a small intestine. So next let us see what happens in the small intestine. The digestion that takes place in the small intestine, we can just list it out here. Now small intestine is the largest portion as we know, and here the digestion is completed, and after digestion the end products will be maximally absorbed also from the small intestine. So the chyme which is received in the small intestine is acted over by 3 juices, that is bile juice, pancreatic juice and the intestinal juice itself. Now the bile juice turns the medium of the chyme into alkaline, and even pancreatic juice and succus entericus contribute to it. The pancreatic juice contains some of the inactive proteases, and these inactive proteases, the most important one is trypsinogen. Now this trypsinogen is activated by enterokinase. Now this enterokinase is an enzyme secreted from the intestinal glands, that is, it is a component of succus entericus. But this is one enzyme which is not acting on any food, but rather it is utilized in the activation of a protease. So trypsinogen is converted into the active form, that is trypsin. And once trypsin is formed, it can self-catalyze the further activation of trypsinogen molecules, as well as it catalyzes the activation of other pancreatic enzymes, that is chymotrypsinogen and procarboxypeptidase. These are activated by trypsin, and as we can see their activated forms, that is chymotrypsin and here it is carboxypeptidase. Now, enterokinase as an enzyme is very important for the question point of view also, because this is an enzyme of the intestinal juice, but it is not acting on any food, but rather it is utilized for activation of inactive trypsinogen. Now, next thing that is happening in the small intestine, we can mention that bile juice of course contains no enzymes, but it has bile salts. And recall the names of the bile salts. Those were sodium taurocholate and sodium glycocholate. They emulsify the fats. And what does emulsification mean? It is that fat being insoluble. So when we take in food, what happens is that fat being insoluble, it will be just floating over the watery surface in the form of a large globule. Now this large globule cannot be acted over by lipases. So this has to be broken down into small tiny droplets, and that's what is emulsification. Now this emulsification is brought about by bile juice as well as the agitation of the intestine, because the intestine is contracting and relaxing the muscularis of it, and these contractions and relaxations are not only the peristaltic movements, but there are segmental movements as well. So that crushes, and so just like when we put something in a mixer grinder and it breaks up into smaller pieces, so that kind of thing is happening, and that's what is emulsification. Now these fat droplets, as soon as small droplets are formed, they are surrounded by bile salts, and these bile salts, when they surround it, this structure is called micelle, and this micelle is a water soluble structure, and we know that for any enzymatic action there has to be aqueous medium, and in aqueous medium only the enzymes can act. So this micelle is a water soluble droplet surrounded by number of bile salt molecules, and now it is suitable for the lipases to act upon. Now these are the various steps which are taking place in the small intestine. Now let us use this information and answer a question. So here we have a question: select the incorrect option. Bilirubin and biliverdin are bile pigments. Now that is correct, which are the breakdown products of hemoglobin. Emulsification is the breakdown of fats into very small droplets. That again is correct. Renin is a proteolytic enzyme found in the pancreatic juice of infants, which helps in the digestion of milk protein. Now we know that renin is a proteolytic enzyme. That is correct. It is found in the infant's digestion of milk protein, but it is part of the gastric juice. It is secreted by the gastric glands. So this one is the incorrect option here. Then fourth one, mucus and bicarbonates protect the mucosal epithelium from excoriation by highly concentrated HCl. That is absolutely correct. So here our answer would be this third one, that is option 3. So this brings us to the end of today's class, in which we have completed with the anatomy of the digestive system and some aspects of digestion. Now in the next class we will be taking up digestion in detail, where we will discuss the digestion of carbohydrates, fats, proteins and nucleic acids. So I suppose whatever we have done today, you will find it useful, and thanks for watching.